Good morning, everyone. Hope everyone is doing fine. Let's begin with today's class. So, as we say in the starting, let's crack NEET PG. Today we are going to discuss about allosteric enzymes and isoenzymes. But before beginning with today's topic, let us have a quick introduction about Unacademy. Unacademy is one of the India's largest online platform for the preparation of examination. You will get the best educators all over India at Unacademy, and you can be highly benefited by their mentorship. There are special classes conducted by Unacademy, and what is so special about these special classes is you can ask your doubts, questions directly with your faculty, and they'll be very much willing to help your doubts to be solved. Then, as I always say, for cracking any kind of examination, it is very much important to evaluate your progress. So that can be easily achieved with the help of test series. There are free test series, free mock tests, which are timely conducted on the Unacademy platform. So you can go there and check in particular topic how much you have learned so far. With the help of Unacademy, in today's time, the learning has become very easy. You can just log into your account and start studying at any time. So there is lots of flexibility in the schedule. No matter how much you are busy in your daily schedule, the classes can be attempted or they can be done at any point of time. So flexibility of schedule is the most important thing. Then language of your choice. There are many faculties who teach in different languages as well. So language of choice is also there. Then as I told you that there are quizzes and test series and videos. So all these things can be done as many number of times as you want to. The quizzes can be retaken, the recorded videos can be rewatched. So it is one of the very convenient way for the preparation of examination. What you need to do for this is you can just directly go to Play Store and type Unacademy, and you will be seeing the logo of Unacademy Learning app. You can download the app and you are good to go. Just make your account and you will enter into the world of immense knowledge. Then don't hesitate to feel like is it something different than normal offline teaching? So it's very much like normal online teaching. You can chat with your educator because there are live classes, unlike the other coaching classes which also post their videos on YouTube. How much it is different from those, how much Unacademy is different, and why is it excelling? Because you can chat with your educator. There are live classes at the time when the teacher is teaching you something. At that time also you can ask your queries and get them solved. And there are interactive polls where you will be needed to attempt the questions, and you will also come to know how much students they are confused in the options, how much the options they are similar. So interactive polls can be highly helpful in understanding how the options they create the confusion, and you will very much feel connected to the class. Then learning anytime, anywhere with the help of Unacademy. So whether you are doing internship or you are already a MBBS pass out, you can just conveniently learn any time, anywhere as per your convenience. Then there is another option, that is the plus subscription. And what is this plus subscription is, by taking this plus subscription, you will be eligible to get all the topics of all the 19 subjects on the Unacademy app. And this plus subscription is chargeable, and the charge is very nominal, and you can just take the subscription and start preparing, right? So for this plus subscription, what you need to do is you need to pay a minimum amount, and in that minimum amount also you can get an additional 10% discount if you are using my referral code. My referral code is FB01. So don't forget to use my referral code and avail an additional 10% discount. And this plus subscription can be highly beneficial for the studies. What is my personal suggestion is go for a yearly or a 2 yearly subscription, because there is immense videos, so you can make the best out of all those available data. So go for a yearly or a 2 yearly subscription. I think we have understood the concept. Now coming finally to the topic, that is isoenzymes and allosteric enzymes. Isoenzymes and allosteric enzymes. Right? This is the topic. We have understood the enzymes and enzyme inhibition in very detail in previous videos, right? So in this video, we are doing isoenzymes and allosteric enzymes. By this, we'll be completing the series of enzymes, because we know what is enzymes, what are the characteristics, what is the classification of enzymes. Then we know about the apoenzymes, holoenzyme, cofactors, coenzymes, prosthetic group. All these things we have learnt in detail. So coming directly to the topic of today, that is isoenzymes. First, I'll be telling you about isoenzymes, and then we'll be discussing about allosteric enzymes. So what is isoenzymes? The first thing, what is an isoenzyme? Which thing will be called as isoenzyme? Right? So what is an isoenzyme? They are different form of same enzymes. Different forms of same enzyme. Right? Different form of same enzyme. What does that mean? That means that they can catalyze the same reaction. Catalyze the same reaction. This is your isoenzymes. As the word itself is telling, iso and enzymes. So how are they different from each other if they are catalyzing the same reaction? How are they different if they can catalyze the same thing? So their origin can be different, or their alleles can be different, the tissue of action can be different. So that's why they are kept into the category of isoenzymes. Right? So formation of isoenzymes we'll be seeing in today's class. We'll be seeing the characteristics of isoenzymes, and then we'll be seeing what is the clinical significance of isoenzymes. Clinical significance of isoenzymes, right? So let's begin. First of all, let's begin with the example of lactate dehydrogenase. This lactate dehydrogenase, it has got 5 isoenzymes: 1, 2, 3, 4 and 5. These all are the isoenzymes of lactate dehydrogenase, and they all catalyze the same reaction. That is, it converts lactate into pyruvate, and the enzyme is lactate dehydrogenase. Another thing that will be participating in the reaction is NAD will be converted into NADH. So this is how lactate dehydrogenase, it performs the conversion of lactate into pyruvate. Right? So all of the isoenzymes of LDH, they are doing the same reaction. They are not performing any different kind of reaction. They are performing the same reaction. So if they are performing the same reaction, where are they different? Or how can you differentiate them from each other? So they differ in the electrophoretic mobility. They differ in electrophoretic mobility from each other. Electrophoretic mobility is different for all of them. It means when they are run in the electrophoresis, they will be moving at different distance in the unit. So they differ in the electrophoretic mobility. Right? So first we have understood the definition along with one of the example. Let's move towards how they are formed. How these isoenzymes are actually formed. This is the next question. So how they are formed, there are various categories on the basis of how they are formed is kept, right? So we'll be dealing each category individually. First of all, there are true isoenzymes. True isoenzyme. Then there is an example of LDH. LDH. Then there are isoforms. Then alloenzymes or allozymes. In a better way, we can call them allozymes. So true isoenzymes, LDH, isoforms and alloenzymes or allozymes. These are the basis on which they are formed. So remember these four categories of how they are formed, their origin, right? So first of all, when we are talking about true enzymes, true isoenzymes, so how they are formed, these true enzymes, take a different color. How they are formed, they origin from different genes, origin from different genes, and these different genes can be located on different chromosomes, on different chromosomes. So true isoenzymes, their origin will be different. On the chromosome which they are present, that will also be different. So one of the example of such isoenzyme is amylase. Amylase. So there are two forms of amylase, that is pancreatic and salivary, and they are present on different genes. These two forms of amylase, they are present on different genes, right? Then coming to the another example of how they are formed, that is LDH. So there are different isoenzymes of LDH, and all isoenzymes of LDH are present in all the individuals of the population. There are different different isoenzymes of LDH. All are present in same individual, and all the individuals of the population, all the individuals of population, right? So we have seen about true enzymes and LDH. True enzymes, different chromosomes or genes we are talking about, different genes for the same enzyme. Then LDH, how is it different? That there are various forms of LDH and all of them are present in a single individual, and all of those forms are present in each and every individual of the population, right? Now coming to isoforms. What are these isoforms? The enzymes are proteins. So when these proteins, which are finally leading to the formation of enzyme, they undergo various different type of post-transcriptional modification. So they differ from each other on the basis of post-transcriptional modification. Differ from each other on basis of post-transcriptional modification. Right? So as we are seeing example of each and every one, what is the example of isoforms which undergo post-transcriptional modification? So the example is alkaline phosphatase. They are different from each other just on the basis of post-transcriptional modification, right? Then comes allozymes. How are these allozymes different from other? So they are the alleles of same genes. Alleles of same gene or a gene make them. So alleles A double L and A double L. This is how you can remember. Now when we are doing the example of each and every one, what is the example of allozymes? The example of allozymes is glucose 6-phosphate dehydrogenase. The example of allozyme is glucose 6-phosphate dehydrogenase. And there are more than 400 types of variants of glucose 6-phosphate dehydrogenase. But how is it different from the other forms that only one variant is present in one individual? One variant, one individual. Right? So we have understood about the origin of these isoenzymes. So true isoenzymes, what was the example? Amylase. When we are talking about LDH, so it is itself a class LDH. They are present in the same individual, many forms, and they are present in all the individuals of the population. Then isoforms. Isoforms, the example is alkaline phosphatase. The example is alkaline phosphatase. And when we are talking about allozymes, its example is glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase. Right? Hope you have understood what I have told you in the formation of isoenzymes. Right? There are 4 characteristics. Now, coming to the characteristics of isoenzymes, or how they are having their properties of these isoenzymes, right? So what are the properties of isoenzymes? First is electrophoretic mobility. The first property is electrophoretic mobility. Let's take another color. Electrophoretic mobility. So we have already discussed about electrophoretic mobility, that which enzyme differs in electrophoretic mobility. Just in the starting of the class, when we were doing the definition of isoenzymes, I told you that LDH, there are many forms of LDH, though most common are 5, and they differ from each other on the basis of electrophoretic mobility. Right? So there are 5 forms of LDH, that is LDH1, LDH2, LDH3, LDH4 and LDH5. They run differently in the electrophoresis. So if they run differently, it means some of them will be reaching faster towards the end and some of them will be reaching slowly, or they won't be reaching till the end. So which one is the fastest amongst all? LDH1 is fastest in electrophoresis, and LDH, here I have written 4. LDH5 is slowest amongst all. So this is how LDH can be differentiated on the basis of electrophoretic mobility. Now coming to another enzymes, that is creatine phosphokinase and alkaline phosphatase. So creatine phosphokinase, they are of 3 types, and alkaline phosphatase, they are of also many types. They can be separated by electrophoresis. So whether creatine phosphokinase and alkaline phosphatase, they can be separated with the help of electrophoresis, or they cannot be separated on the basis of electrophoresis. So yes, these 3 classical examples of isozymes, they can be separated on the basis of electrophoretic mobility, right? Then second characteristic is stability of heat. that how are they stable in heat. So there are heat labile isoenzymes and there are heat resistant isoenzymes. So alpha 2 is a kind of heat labile enzyme, heat labile, and this form is present in liver. So stability of heat, some of them will be stable and some of them will be non-heat stable. So one of the example of non-heat stable is alpha 2 and this variant is present in liver, right? Now coming to the third characteristic, that is there are various inhibitors of the iso enzymes. So some of the inhibitors, they completely stop the reaction. One of the classical example is tartrate labile acid phosphatase. Tartrate labile acid phosphatase. So the inhibitor in this case is tartrate, and when tartrate reacts with acid phosphatase, it will just inhibit the reaction, right? Then coming to fourth characteristic, that is cofactors. So cofactors, one of the example for this is isocitrate dehydrogenase. Isocitrate dehydrogenase. What happens in this is mitochondrial variant, it needs NAD, and cytosolic variant, it needs NADP. So the cofactor which is required by isocitrate dehydrogenase is different on the basis of its position. If it's a mitochondrial isocitrate dehydrogenase, it will need NAD. And if it's a cytosolic variant, it will need NADP. Right? Now coming to the fifth characteristic, that is sensitivity to the antibodies. So various isozymes, they differ on the basis of antibodies. Like there can be 10 types of isoenzyme for a single enzyme, and some of them will be sensitive to antibodies and some of them won't be showing any sensitivity towards the antibody. So this is also seen in the case of isoenzymes, right? So 5 characteristics we have seen so far. Let's move forward. The sixth characteristic is origin tissue, the tissue of origin. So we have seen there are various characteristics which depend on the tissue in which it is originating or in the tissue where it is showing its action. So one of the example that I've included for the origin of tissue is alkaline phosphatase. This alkaline phosphatase, what happens in this is there are various tissue of origin for alkaline phosphatases. So some of the tissue of origin I'll mention here is bone, then liver, then intestine, then biliary, placental, and leukocyte. So they can be differentiated on the basis of the tissue in which they are originating. So on the basis of tissue in which they are originating. So one of the example we have dealt over here is alkaline phosphatase, and there are various categories of alkaline phosphatase on the basis of the tissue they are developing. The examples of the tissue where they are developing is bone, liver, intestine, biliary, placental, and leukocytes. Right? Now the last characteristic of isoenzyme that we need to see is Km value and affinity. So Km value and affinity, how the Km value is related to affinity? Km value is inversely proportional to affinity, right? Affinity of enzyme towards its substrate. This is the affinity, how the enzyme is getting attracted towards the substrate. This falls under the category of affinity. So this affinity is used for substrate, not for inhibitor or modulator or anything, right? So how this Km value and affinity, they change in each case of isoenzymes. So for each isoenzyme, the Km value will be different. Km value is different. So always remember that Km value can never be same for different types of isoenzymes. Okay. So these are the characteristics. So what are the characteristics we have seen? Electrophoretic mobility, stability to heat, inhibitors, cofactors, sensitivity to antibodies, then tissue of origin, and then Km value and affinity. These characteristics we have seen. Now we'll be dealing some of the examples of these isoenzymes, and the examples which I have included in today's class is LDH, CK and AP. LDH, our lactate dehydrogenase, lactate dehydrogenase, CK, our creatine, phosphor, kinase, and AP, alkaline phosphatase. Right? So we'll be dealing with these examples. Right? So first example that we are going to discuss is LDH. So since the starting of the class, we have been saying that there are various variants of LDH. So how many we have seen till far? We have seen it's 5 types. So let's write down LDH 1, 2, 3, 4 and 5. So this LDH enzyme is made up of tetramer. Tetramer of 2 types. It's a tetramer made up of 4 chains of proteins of polypeptide, right? The tetramer made up of 4 chains of polypeptide. LDH1, the chain in LDH1 is of one type only, that is H. For 2, there are 3 chains of H and 1 chain of M. For LDH3, it is equal number of both chains H2, M2. LDH4, 1 chain of H and 3 chains of M. For LDH5, there will be 4 chains of M. Right? So these variants, they are present in different tissues as well. LDH1 is present in heart. Then LDH4, it is present in liver. And LDH5, it is present in muscle. So this is the place where it is found, and this is the characteristic of the protein chain or polypeptide chain each LDH is containing, right? The LDH4 or the LDH1, it contributes 27% of total LDH, and LDH2, it contributes to 35% of total LDH. So this is how the LDH is differentiated on the basis of the number of chains and on the basis of where it is present, and lastly, what is the amount of each LDH in the body, right? Now coming to its importance, let's take another color. In myocardial infarction, in MI, what happens is the concentration of LDH1 increases. Concentration, it increases. Otherwise, its concentration is low. So, this is also called as flipped pattern. As you can see, it is 27% previously. But in case of Mi, the pattern will be flipped. So this is called as flipped pattern of LDH. What happens in this is the LDH1, it starts increasing in case of Mi. And when does it starts increasing? 6 to 12 hours after Mi. So if it is 6 to 12 hours later, it means it's a late marker. Here we can write it's a late marker of Mi. Late marker of Mi, because its concentration it starts increasing 6 to 12 hours after. Then it's on peak when 24 to 48 hours are reached after Mi. This is the peak of LDH1 in case of Mi, right? And in some of the cases, you will also see that the peak is attained in 3 to 4 days, right? And its concentration comes to normal in 8 to 14 days. In 8 to 14 days, its level will be normal. So, this LDH, it's a late marker of Mi. It's a flipped pattern of LDH because previously its concentration will be lower, and when Mi has occurred, it will lead to increased concentration. And why it is called as late marker? Because its concentration it increases 6 to 12 hours after Mi. The peak is attained, can be said, at 2 and 3 days, and its normal level is attained after 8 to 14 days. It takes 8 to 14 days to come back to normal, right? So we have discussed everything about LDH. Our first example. Right? LDH. Now let's move towards second example, that is creatine phosphatase CK. What is its reaction? Like for LDH, we have seen the reaction in the very first slide only, that what it does. In the very first slide, we have seen that lactate is converted into pyruvate with the help of lactate dehydrogenase. So we just did not repeat it in the last slide. Here, when we are discussing creatine for the first time, so what is the function of creatine phosphokinase? What it does is, it converts creatine into phosphocreatine. So we know that kinase word, whenever it is included in the enzyme, it is responsible for transfer of phosphate group. So here also creatine, what is it doing? Creatine phosphokinase, what it is doing? It is transferring the phosphate group, right? So when we were discussing about LDH, we were discussing that how many polypeptides make up a single LDH. And when we are talking about creatine kinase, so how many chains are there? So it's a dimer. This CK is a dimer, right? So the chains, they are of two types, M and B, and CK itself is of three types, CK1, 2 and 3. CK1 is made up of two B chains, 2 is made up of one M and one B chain, and 3 is made up of 2 M chains, right? So this is the difference on the basis of polypeptide chains. Now, when we were discussing LDH, we also saw that it is present in which tissue and all. So here we will be seeing that its concentration increases in which kind of disorder. So CK1, it increases in brain infection. CK2, it increases in myocardial infarction. And CK3, it increases in muscular dystrophy. Right? CK1, it increases in brain infections. Then CK2, it increases in myocardial infarction. And CK3, it increases in muscular dystrophy. Right? So this is how it is done. Now, if you can see over here, the CK2 increases in myocardial infarction. So when we were talking about LDH, there also we have seen similar thing, but it increases after some time. So if it increases after 6 to 12 hours, it was called as late marker. The CK2 is the earliest marker of MI, the earliest marker of MI. Very important. And when does they increase? This enzyme concentration, when does it increase? It increases 3 to 6 hours after it increases, right? Just after the attack, it will start increasing and it will be respectable at 3 to 6 hours, and then the peak is attained at 12 to 18 hours. 12 to 18 hours peak is reached, and it becomes normal in 36 to 48 hours. Normal. So CK2 is very important in diagnosing MI. Very important diagnostic enzyme. Very important for diagnosis of MI. So remember this thing. So we have discussed our CK, right? We have discussed about creatine phosphokinase, and it transfers the phosphate group from creatine to phosphocreatine. The molecule creatine is converted into phosphocreatine. It's a dimer made up of two chains, that is B and M chain. CK1, it increases in brain infarction. CK2, it increases in myocardial infarction. And CK3, it increases in muscular dystrophy. And CK2 is the earliest marker of myocardial infarction, and LDH and your LDH1 was the late marker of myocardial infarction. So we have discussed these two enzymes in detail. Let's move to the third example of our isozymes, which we need to discuss in today's class, that is ALP. So ALP, its full form is alkaline phosphatase. Alkaline phosphatase. So what is it? Alkaline phosphatase is, there are 6 different isoenzymes of alkaline phosphatase. 6 isoenzymes of alkaline phosphatase are there. And how does they differ? They differ with each other on the basis of composition and the tissue of origin. Composition and tissue of origin. So let's have a look on this. Alpha 1, alpha 2, and alpha 2 is of two types, then pre-beta, gamma, and LAP. These are the categories of alkaline phosphatase. So alpha 1, where is it present? It is present in biliary tree. It is present in biliary tree. Alpha 2, it's also your heat labile form, and it is present in liver, and it contributes the 25% of activity. Alpha 1, it was contributing to 10% of total activity. Alpha 2, another form, is heat stable, and this heat stable is present in placenta, or it's the placental alkaline phosphatase. Pre-beta, it is present in bone and it contributes to 50% of activity. Then gamma, it is present in intestine, and LAP, it is present in leukocyte. So alkaline phosphatase, their composition is different. That is the chains alpha 1 chain, alpha 2, and alpha 2 itself is of two types, heat labile and heat stable. Then pre-beta, gamma and LAP, and how much they are contributing to the total activity. Alpha 1 is contributing to 10%, alpha 2 heat labile is contributing to 25%, and pre-beta is contributing to 50%. So this is how the alkaline phosphatase is categorized. Now coming to the clinical importance of alkaline phosphatase, which is quite important to know. So coming to clinical importance of alkaline phosphatase, right? So first thing, take another color and if possible another slide. I don't have, okay, fine. Let's take another color. Let's go. So alkaline phosphatase, its activity is high in kids. Activity is high in kids and growing adults or growing kids, high in kids and growing individuals, and its activity is higher in hepatitis as well. Its activity is high in hepatitis. So the activity, it increases how many folds? 2 to 3 folds of the normal activity. And how much it is present? It is 30 to 300 international unit per liter is there. Then second, the activity increases 10 to 12 folds in obstructive hepatitis. Its activity increases 10 to 12 folds in obstructive hepatitis. And it's a kind of ectoenzyme. Third, its activity increases 25 times. This, let's write times or folds for each. 10 to 12 times, 25 times. Its activity increases 25 times in bone diseases. Fine. Then there are other things also which are related with clinical importance of ALP. That is Regan isoenzyme, that is the carcinoplacental variant of ALP found in carcinoma of lungs, liver and gut. Right? Regan isozymes of carcinoplacental variant of alkaline phosphatase. Then Kashara isoenzyme is found in fetal intestine and it's a variant of ALP. So I'll just write the names, won't be writing the whole thing. So Regan isoenzyme, then Kashara isozymes, and lastly Nagao Isoenzymes. They are the variant present in the germ cells. This Nagao is in germ cells. Germ cell variant. Germ cell variant of ALP. And Kashhara is the fetal variant. Fetal variant. And Regal isozyme is the carcinoplacental variant. Carcinoplacental variant. So, alkaline phosphatase is also one of the very important isoenzyme according to exam point of view. Various questions are being asked on this also. So, just keep in mind all the things that we have discussed about LDH, CK and ALP. Fine. Let's move further to the next part of today's class, that is allosteric regulation. We'll be discussing about allosteric regulation. So what is allosteric regulation? So regulation of enzyme quality, that is the intrinsic quality of the enzyme, comes under allosteric regulation. Now there is another type of regulation also, that is the quantity of enzyme. How much enzyme will be produced. So that is regulated by induction and repression of particular gene, and then enzyme degradation. Every enzyme will have its own half-life. So if an enzyme is produced, for how much time it will be there. So that will be determined through enzyme degradation, and how much it should be formed. That will be covered under regulation of enzyme quantity. The first thing, that is regulation of enzyme quality, is called as the intrinsic catalytic efficiency. It is of two types, allosteric regulation and covalent modification. Right? Allosteric regulation and covalent modification. Now, in today's class, we are talking about allosteric regulation. So, allosteric enzymes, they have a catalytic site where the substrate bind and a separate site for a modifier which bind. So let's draw a structure of these allosteric enzymes. So maybe like this can be the structure, and this is your substrate, and this is your allosteric modulator. So this is substrate and this is allosteric modulator, and this is the enzyme. So substrate will be binding at other place, and allosteric modulator, it will be binding at other place, right? So substrate will be binding at the active site and allosteric modulator will be binding at some other place. And when allosteric modulator will bind at some other place, it will lead to change in the conformation of the active site. So now your substrate would be binding to the active site more tightly, or it will be binding loosely. So if a modifier or an allosteric um, modulator is an inhibitor, so it will stop the reaction. It will change the active site in such a way that now no substrate can bind over there. And if a modifier is an activator, it will change the active site in such a way that now the substrate can bind more tightly with the active site. Now, what are the features of allosteric regulation? The modifier need to be structural analog of the substrate. Need not to be the structural analog of the substrate. If it is different than the substrate, so yes, they are occupying different places. So obviously they can have different type of structure, different type of shape. A similar type of shape is not that much necessary because they are occupying different place, need not be structural analog. Partially reversible when excess substrate is added. So yes, these allosteric regulation, they are of non-covalent type. The bonding that occurs is non-covalent bonding. So what all comes under non-covalent bonding is your ionic bonds, and your van der Waals force. So these type of bonding will be there between the enzyme and allosteric regulator, and the binding will be reversible. The binding between the enzyme and modulator will be reversible. So how is it reversible? If the substrate is in large amount or the pH or the buffer is changed, it will lead into reversible binding, right? So when excess of the substrate is added, the reaction will be reversible. Then most allosteric enzymes have a quaternary structure and made up of subunits. So these allosteric enzymes, they are not made up of a single polypeptide. They are made up of multiple polypeptides. They have a quaternary structure and made up of subunits. The allosteric enzyme themselves will be made up of various polypeptide chains, and the most common number of polypeptides is 4. Right? Allosteric enzymes, they occupy the key regulatory position in the metabolic pathway and called as key enzymes or rate limiting enzymes. Very important. Rate limiting enzymes, we always talk about the rate limiting enzymes. So they are the key enzymes and rate limiting enzymes. So rate limiting enzyme, what does this rate limiting enzymes means? What is this rate limiting enzyme? Which enzyme will be called as rate limiting, or which enzymatic process will be called as rate limiting? Those enzymatic reactions which are slowest are called as rate limiting enzymatic reactions, and these rate limiting enzyme reactions are catalyzed by allosteric enzymes. Most of the times they are allosteric in nature, and thus they are playing a very important role in metabolic pathways. Fine. Hill's equation describes the behavior of enzymes that exhibit cooperative binding of the substrate. So, in the next slide, I'll be telling you this. Sigmoid shape of the curve is due to cooperative binding. Let's see this in detail. See, before moving with the types, let's see this. There will be cooperative binding. That means if there are 4 polypeptides in a particular enzyme, it means it will have 4 catalytic sites if all polypeptides are same. So once they have binded with one kind of substrate, it will show high reactivity towards the coming substrate. Its reactivity, its acceptability, its affinity will keep on increasing. This is what is explained by Hill's graph or Hill's equation, and the shape of the graph is sigmoid, right? So they show cooperation in binding. They will show cooperation in binding, right? Then there are two types of allosteric enzymes, that are K series and V series. K series and V series. So what are these K series and V series? Let's see them in detail. Let's take the pen. So what happens in K series? In K series allosteric enzymes, Km value increases and V max remains same. And the in V series, what will happen? V will increase and Km will remain same. So the name is given on the basis of what it is altering. It is altering Km, and in both of the cases there is always rise. So in K series, it is related to the Km value, and when we are talking about V series, it is related to the Vmax value. Vmax is increasing. So what is this Vmax and Km? If you people can remember the Michaelis-Menten equation, what is it? It is V0 is equal to Vmax into S upon Km plus S. So if Vmax is increasing, how the graph will be changed? Let us take another color. If it is becoming Vmax, how the things will be changed. Let's draw a graph, a small graph over here. If see, this is your normal Michaelis Menten, and if Vmax is increasing, it means it's a positive modulator. So it will become like this, and if there is a negative regulator, the graph will become like this. So in all of the cases, there is no effect on the Km value, but your Vmax is increasing, right? And if we draw a graph for K series, how will it be different? Will be like this. When there is a positive modulator, and for a normal Michaelis Menten graph, it will be like this. And if there is a negative regulator, it will be like this. So in all the three cases, in K series graph, the value of Km is remaining, the value of Km is increasing, and in V series the value of Vmax is increasing, and the graphs, they are related to the Michaelis Menten equation or Michaelis Menten graph. Michaelis Menten equation oblique graph. So I hope you people have understood that how the things are occurring in the case of allosteric regulation, right? So we are done with isoenzymes and allosteric enzymes, and we have discussed all the important points which are important according to the competitive exam point of view. Now lastly, let's have a little comparison of allosteric regulation with non-competitive inhibition and similarities and how they are different. So modifier or inhibitor is not a structural analog of substrate when we are talking about allosteric enzymes. Why? Because they occupy different place. The modifier and inhibitor bind to a site that is distinct from the substrate binding site. When we drew the structure, we drew a different place for the binding of the modulator. And V series allosteric enzymes and non-competitive series of allosteric enzymes and non-competitive inhibition, and Km remains same and Vmax decreases for non-competitive inhibition. So that is it about the theory. Let us have a small look on how the questions will be asked when there will be questions on the basis of the isoenzymes and allosteric enzymes, right? So some of the examples that I have included over here is, let's move. LDH5 level increases in, so increases in which cell injury? So LDH5 is present in muscle, so its concentration will increase in muscular injuries, right? Because it is a variant which is present in muscle. So the variant of liver is your LDH4, your variant of heart is LDH1, your variant of muscle is LDH5, and your variant of RBC is LDH3. So this is how the things they occur, right? So 5 will increase in which type of cell injury? Muscular cell injury. Now, which of the following LDH is having the fastest electrophoretic mobility? So, we have seen that LDH1 is having the highest electrophoretic mobility, and the slowest amongst all is LDH5. LDH5 is slowest and LDH1 is fastest in electrophoretic movement. Now, coming to the next question. Which of the following enzyme is not raised in liver disorders? So, the enzymes which are not raised in liver disorders are lipase and urease. Otherwise, alkaline phosphatase, this will increase in liver disorders. Now, coming to the next question. Which of the following estimates blood creatine level most accurately? Which of the following estimate will do the best check of the creatine level? So, enzymatic activity is the best assay for checking the blood creatine level, right? Now, what is true about isoenzymes? Which of the following characteristic is true about isoenzymes? They catalyze the same reaction. Is it so? So yes, they catalyze the same reaction, same quaternary structure. So cannot be said. Same distribution in different organs. No. Same enzyme classification with same name and number. So no. The thing true about enzyme is they catalyze the same reaction and they cannot share the same quaternary structure because they are showing different electrophoretic mobility. So the only option which is true amongst all is they catalyze the same reaction, right? So this is it for today's class, and we have almost completed everything in isoenzymes and allosteric enzymes. And in previous videos, we have discussed everything about the introduction of enzymes, classification, their nomenclature, holoenzyme, factors affecting, then theories for the enzyme action. We have discussed almost everything. And in another video, we have discussed about the enzyme inhibition, cooperative, competitive, non-competitive, uncompetitive, suicide, and feedback inhibition. So this is it for enzymology part. We have tried to cover everything in the series. So go and check out the other videos also. And lastly, thank you very much for your patient listening. Before leaving, just don't forget to like my video. It really motivates me. And also subscribe the channel Let's Crack NEET PG. And for getting all the notification, hit the bell icon for getting all of the upcoming videos by other faculties as well as the videos by me. Also, if you are going for a plus subscription which can be highly beneficial, use my referral code, that is FB01. So have a very good day, happy studying, and we'll meet again next time with some other interesting topic of enzymology. So that is it. Have a very good day. Bye.